Well, I'm going to argue that DNA is absolutely integral to the process of cancer, in that cancer is largely a disease of damaged DNA. It arises because of changes in our DNA, either in result to things like environmental carcinogens, such as tobacco smoke or ionizing radiation, or things we eat in our food that are not good for us, or it can arise because we inherit damaged DNA from our parents. In other words, DNA that has changed slightly in ways that make our cells misbehave.
And I guess, how common are the cancers then that are caused in this way? So you've mentioned carcinogens such as smoking there, which is obviously then lung cancer. But how prevalent are these various forms of cancer?
I would argue that most forms of cancer involve changes to our DNA. The vast majority of cancers are caused by subtle changes to our DNA. Not just any old bit of DNA, but particularly two types of DNA encoding two types of activity. One you can think of as the accelerator of a car. It's like jamming the brick on putting a brick on the accelerator so the car keeps on going when it should stop, and our cells keep on dividing when they should stop. The other class are a set of genes which stop our cells from dividing when they shouldn't, as our cells should only divide in the right place at the right time. And so changes to these bits of DNA are the equivalent of cutting the brake cables or the brake pipes so that the car can't stop when it comes against an obstacle. And if we inherit changes, mutations in our DNA from parents to these types of gene, then we have a greatly increased risk of cancer.
And I guess, what examples of cancer then have these mutations?
Well, there are a large number of examples we could give. Two of the best known would include an inherited form of blindness, retinoblastoma, in which a gene which should serve as a brake telling cells not to divide until the right signals come has been mutated so that the patient inherits a damaged copy of that gene from one parent, and then a subsequent change in another copy, in the residual copy of the gene from the other parent, will result in cancer. That's one well-known and well-studied case. But another would be cases of inherited breast cancer, in which some of the inherited predispositions to breast cancer are, again, inheriting a damaged copy of a gene which should protect the body from breast cancer. But if a damaged copy is inherited from one parent, again, it dramatically increases the risk of breast cancer arising.
And I guess, well, this just shows just how integral DNA really is to cancer, as you're arguing. But as well as actually causing cancer, the interesting thing is that DNA is also what's targeted to treat cancer.
Absolutely. That's a fascinating paradox, that although cancer is caused by damaged DNA, it's also treated by inflicting further damage on DNA by two of the three main forms of treatment. Obviously not surgery, which doesn't do that, but radiotherapy is aimed at damaging DNA of the cancer cell, and many forms of chemotherapy are also aimed at damaging the DNA of the cancer cell. This seems perverse when DNA damage is a cause of cancer, but the rationale is that a cancer cell, in order to become a cancer cell, has already jammed a brick on the accelerator and cut through the brake pipes.
Yeah.
So therefore, if the cancer cell comes across a roadblock which says stop, a normal cell will gently grind to a halt and wait until the damage is repaired, and then continue to copy its DNA. The cancer cell can't do that. It's given up that option by becoming a cancer cell, so it crashes catastrophically and dies. So it's a means of selectively killing the cancer cell by overloading it on DNA damage.
And it's proven to be very effective.
It's very effective, sadly not quite effective enough, because cancer, of course, still kills a quarter of the population in the UK, and it affects a third of us. So there's still room for improvement, but those treatments are at least effective in prolonging life and in some cases in curing cancers.
And I guess the important thing, though, with cancer, a real kind of benefit to treatment of it is when it's caught early and diagnosed early, which is what your research kind of is hopefully providing a method of.
Absolutely. I think in many ways you could argue that the most effective thing we can do to combat cancer at present is to catch it earlier, because then the existing treatments become more effective. Inventing new treatments is happening, and it's happening quite effectively, but progress is inevitably rather slow. Clinical trials take a lot of time, and not all treatments that look good in the early stages stand up to scrutiny in the late stages. So therefore, if we can catch it earlier, the existing treatments that we've already developed can be that much more effective. And so we've focused on trying to identify precancerous and cancerous cells in body fluids from a range of tissues, such as a cough test for lung cancer, coughing up sputum and looking for precancerous cells there, or bowel cancer, or in bladder cancer, or indeed trying to improve the reliability of the cervical smear program.
Well, this is your area of research, really, and you're focusing on proteins involved with DNA replication.
Exactly.
As a way of diagnosing or finding cancer cells early. How is this possible?
It's because proteins involved in making DNA are broken down in mature cells that are about to die and be sloughed off into the body fluids, whereas they're present in cells that are dividing or preparing to divide. So most of the cells in our body are not doing either of those things. They're quiescent. They've specialized and they've stopped dividing, or they're simply waiting to divide at some later date. But cancer cells keep on dividing. They're locked in the division cycle. So they have these, or essentially all cancer cells have these proteins that we call MCM proteins. And therefore they're very, very good markers to detect precancerous or cancerous cells very early in the course of the disease.
So this seems like it would be a very clear way to find them. So what stage is this at now? Is it hoped to be in clinical use soon?
There have been clinical trials for several types of cancer. By far the most advanced are for improving the smear test for cancer of the cervix of the uterus. And those tests it's going through multi-center trials, mostly in the USA at present, by Cancer Research Technology, which is the technology transfer arm of Cancer Research UK, has licensed the patents to a multinational company, Becton Dickinson, and they've been conducting multi-center trials when we're very encouraged by the results of those trials, which will go for regulatory approval, hopefully, later next year.